Hello, my name is Cannon Collins. I decided to do prions, so the pesky prions and the roles they play on the translational stage. And the introduction begins with, identifying what a prion is. It's not really a cell, it's not really anything else more than an infectious protein that infects different forms of organisms. Transmission can happen in a multitude of different ways, and we will discuss more on it later. The abnormal folding of proteins is what this prion is known for. It will infect someone's brain and cause a severe neurodegenerative disease. And lastly, prion proteins are still mysterious. They're very understudied and they're very under-researched. This is due to the fact that they don't infect humans as much as viruses do. Now that we kind of understand what a prion is, structure should be what we understand next. And as you can see in the picture, you can see on the left a normal prion and the sheets flowing normally and everything's fine. And then on the right, you can see the disease prion. And these beta-pleated sheets are multiplied by at least tenfold. And this is where the disease comes in. And so this also helps in the beginning of the prion. This infectious disease begins at the secondary stage in these beta-pleated sheets and alpha helixes because it shares the same primary structure as the PRP protein in your head. And then where does this come from? Disease prions can come from a variety of different species and different biological niches around the world. So as we go from structure, we move into function. The functions and structures are just as mysterious as each other. There's no concrete evidence of the function of a prion, but as time has gone on, scientists have hypothesized that due to the highly conserved nature, this was a beneficial protein that entered our body, and it helped stimulate our central and peripheral nervous system to maybe give us a more aware environment. The gene regulation of these is pretty mysterious as well. These phenomenal amino acids can change the conformation states of these proteins that they infect at the whim of whenever they want. So if they're in a bad environment, they can change into a more stable state of the prion. And then lastly, the translational factors. Translational factors are affected by prions usually through the stop codons. In this case, prions can use the stop codons to control how many prion proteins are going to be made in a certain time. Prions bring many problems to the table, especially with such powerful structures and functions. Some of these problems infect organisms such as sheep, as you can see in the bottom left-hand corner. The sheep has scrapie, and it has a death warrant. In the top left-hand corner, the Creutzfeldt-Jakob disease also comes with a death warrant. This brain is completely shut off, and it has almost completely degraded. And then finally, you have the mad cow disease, which you can see in the bottom right-hand corner. It turns off motor functions in cows and switches the temperament from usually a docile temperament to a very aggressive temperament. Prions are very problematic in the fact that their transmission is just as simple as their disease is lethal. Simply eating the meat of a cow infected with mad cow disease will give you Creutzfeldt-Jakob disease. And then while you're infected, scientists will try to target these very small, short amino acid sequences. And doing this is very difficult, and to try to break these down will usually kill the host to put them in that kind of environment. And lastly, the transmission factor used. You can see in the picture right here, the beta-pleated sheets are now infected with the new monomer that will generate new infection cells. So once you're infected with a prion, the protein folding of the prion will follow that of protein synthesis before and gene regulation, transcription, translation. This will start at secondary structures though, because the PRP found in your head and the PRP that's found in the prion share the same primary structure. A basic introduction into the transcription of prions is that prions use these transcriptional regulatory networks, or TRNs, and through these networks are able to target hundreds of transcriptional factors. While these highly lethal prions are alive, how can we study them? Well, we use organisms like mice, E. coli, and different sorts of organisms because these proteins are highly conserved, as mentioned before. And lastly, seeding. This is where an infectious particle will attach itself to a host, causing more infectious seeds to come out, as you can see in the picture in the top right-hand corner. Translation of prions is one of the most interesting and vivid topics of prion diseases. As you can see in this mRNA, this is in the cytoplasm, and these amino acid sequences are ready to be folded into proteins by the chaperone enzymes. The restriction enzyme will cut them, and the chaperone will begin to fold them, and the prions will begin to generate catalytic subunits that lead to mRNA instability and protein degradation. So, what happens after the disruption of translation factors? Well, after the disruption of translation factors, this is where the disease can really propagate and generate more and more prionic cells. So, this is where the disease will look more like a lytic cycle instead of a lysogenic cycle. And then lastly, tying it to what we learned, another connection that these diseases have is the uh, amyloid of a sub-35p, a subunit of the translational termination factor. These factors tell the mRNA when to stop, and these prions will actually invade these stop factors and keep replicating prionic cells to make more prionic cells to infect other cells or just to destroy the cell that they are living in. This is what ties in what we learned in class to what I taught myself recently. Translation happens in the 3 steps, as we all know, and a lot of these errors happen post-translationally, but these errors only happen once post-translationally. Afterwards, they become embedded in the DNA and the codes and then the protein synthesis, and this gets translated over and over and over again. And eventually, in translation, it won't stop due to the mRNA poly A tail being shortened due to the catalytic subunits. This is again tying what we learned in class to what I taught myself. Gene regulation is a huge step in prion diseases and understanding them. And so gene regulation starts with transcription and about how these prions can invade cells with minimal to no resistance, and even when they do get caught by the body, they're really hard to denature and target. So this makes it really easy for a C-amp bind and activates kinase, which travels into the nucleus and phosphorylates the Krebs proteins. This will kickstart protein synthesis and lead into prion propagation. Another key factor of gene regulation is regulation motifs. These are simple membrane-bound proteins that are found throughout history in different organisms in different niches in different biological parts of the world. And then the regulation changes made when infection sets in, you can see that the pathogenic mutations associated with inherited forms of prion disease cause non-conservative point mutations between residues 178 and 232. This inserts 168 base pairs to and this is linked to Creutzfeldt-Jakob disease. And then point mutations are also made between 90 and 150, and these are linked to Gerstmann-Stahlheiser-Schneidner phenotype with large number of amyloid plaques. And then lastly, the influential codon of 129 is a polymorphism, also shown in blue. Prevention and treatment. Prevention is simply don't eat monkey brains, and treatment is there's no real treatment for a prion disease just yet. Lastly, these are my references. I hope you enjoyed my presentation.